Hi, Mike Dolinski with Taurus.ag. Just want to take some time to talk about grain filling. Before the summer is done, your cereals will be filling their grains or their seeds in the spike or in the head. It just so happens we have a product called grain fill, and I'll briefly touch on it later on. But first of all, I want to talk about the process of grain filling and then show you some slides about the actual way that the plant fills and show you some photographs of that I've taken over the years. Now, grain filling refers to the process of cell proliferation and expansion. We have to actually make a cell and then we got to expand it so we have a good sink or a good place for nutrients to be stored away. And then photosynthate processing. In other words, the plant will try and remobilize whatever it produced through the summer in order to fill that seed. And it's usually filled up with starches and proteins. Now, grain filling is the final growth stage in the cereal's life cycle and key in determining grain weight, grain composition, quality, and yield. We need lots of space and we got to fill it. The bigger the seed, the more yield you get, and we got to make sure we've got the nutrients in there that will support what the plant wants to do. It wants to grow a seed to keep its progeny going. We want to eat the seeds because we need the same nutrients that the plant does. So it's a win-win situation. The plant does it for us, thankfully. Now here's the typical growth curve, and we've grown this plant up until grain filling right here, grain filling. It's after pollination. So once we have pollination, we start growing the seed. The seed starts to grow. And we're gonna feed it and we're gonna fill it, and then after that we're gonna ripen it and we're gonna harvest it. Now the way I look at filling is that the seed is now the sink. It's the sink for everything because it's late in the plant's life, and it's the sink for all the nutrients, all the minerals, and so on that the plant has. Now, throughout the life of the plant, there's a source-sink relationship. Now, the source is the assimilate from photosynthesis, which is made from sort of these things, along with, you know, good agronomy. So those sugars are then sent to the sink. And I'll give you an example. A root is a sink because a root can't feed itself, so the shoot has to send nutrients to it. So it's a sink. More simplistically, you know, sources are from photosynthesis, sugar, and from remobilization. The source can be remobilization of the nutrients that are somewhere in the plant, and the plant can move it. So that becomes the source and it moves to where it's needed. So things like seeds, for example, can be both a source. The seeds are the source of nutrients for the seedling. At the same time, the seeds can be the sink at the end of the cycle. So seeds start out as a source and then wind up as a sink in the plant. But primarily leaves are the source. That's where most of the photosynthesis takes place. Photosynthesis takes in the stems. And even old roots can be a source at the end of the day because the plant will try and move everything out of the old roots up into the seed as well. Now the sinks are the growing point and the storage cells. So new roots, new seeds, fruits, and tubers. So I think you get that idea. So now that we're talking about grain filling, we need a big sink and we need a good source. If we're short of one of them, we're not going to get the yield that we want. So what are the storage components? They're primarily starch and protein. Now we know that photosynthesis gives us sugars and that those sugars are used to make the carbohydrates and the amino acids along with nitrogen and sulfur right there. So you want to keep photosynthesis going on in the plant as long as possible. And it's generally the lack of nitrogen at the end of the day that slows and eventually stops photosynthesis. When the plant is short of nitrogen, it'll start, you know, scavenging everything, and then when the plant's sort of running out, largely, we're done. We're done. The seed better be filled. Now here's a cell, and these are the components, the nitrogen components in the cell. And this is a C3 leaf cell, which is basically all the crops except corn. Corn is a C4 crop that we grow in Canada. Now you can see here that this is just a breakdown of the nitrogen in the cell. The chloroplast makes up 75% of the nitrogen. So that's the ribisco, which is the enzyme that breaks down CO2 and allows the plant to produce sugar from the carbon. We got ATP, we got the light, photosystem 2 and the light enhancing molecules that are there. And then we have a sort of a batch here of 16% that is multiple kinds of things, but it's all where the nitrogen is residing. So 100% of the nitrogen in there is almost available for remobilization, except from the cell wall, where about 10% of the nitrogen is tied up. Now, to do that, the plant has to break down all of these molecules, including the vacuole, liquefy them, move them through what are called companion cells from the leaf into the phloem so it can be moved up into the seed, and that's exactly what it does. The enzymes break all this up. Now, there can be problems. If you get heat, the enzymes are damaged. If you don't have water, the enzymes are damaged. If you don't have water, you can't move a lot because the stomata are closed. And even though it's not by transpiration, if you're not getting transpiration because the stomata are closed, your phloem isn't moving things a whole lot either. So stress is really key in terms of giving you that big seed and having it full of nitrogen, amino acids, and basically storage proteins. And here we just see chlorophyll, and you can see that there's 4 nitrogen surrounding the magnesium, and we always talk about magnesium, but there's a lot of nitrogen in chlorophyll. So all of that has to be broken down. Now there are 2 main sources of carbon or sugar filling in cereals: the current production in photosynthesis, and when you have pollination, the flag leaf is really running photosynthesis, and the flag leaf produces the majority or about half of the photosynthates for filling. And by the time the seed is about half grown, the flag leaf is starting to sort of run out of steam, and a little shot of nitrogen at that time may be able to prolong it a little bit and enhance that period of starch sugar production. I should also point out here that after photosynthesis is done, the senescence will start moving in, and carbohydrates will be moved to the seed. It's just a matter of process, and that the stem is about the last reserve there because the plant has to use the stem in order to move all the lower nutrients up into the seed. Now, one of the keys is that the seed has to make space, and after anthesis, when the embryo has been pollinated, the tiny seed starts to grow, and the endosperm cells, or the amyloplasts, have to be built. So the plant moves a lot of nutrients up into that, and there's not much formation to it. It's just juice getting ready to form those amyloplasts, and then we have to fill it with starch granules and blend in the proteins. Now, the starches that are now being used are sugars. Well, let me go back. Starch is just a bunch of sugar molecules that hook together. Now, the starches in wheat are either amylose or amylopectins. Amylose breaks down slower than amylopectin, so that when the seed is germinating, it has a supply of sugar over a longer period of time. At the same time, the plant is blending in proteins into the endosperm, so that the plant, the seedling, the new seedling coming from that seed, will have all its nutrients that it needs, including minerals. The other component is nitrogen. Nitrogen has to be moved, and it's moved primarily as amino acids. And the supply coming in after anthesis usually increases if you can give it a little shot at that time, and that's where grain fill comes in. If we give it a treatment with a little bit of nitrogen, sulfur, and so on, it can help boost it, and I'll talk a little bit more about that when I talk about grain fill. And then the rapid senescence, and I think I mentioned this, is due to nitrogen shortage. It starts running out. The other thing is most of the nitrogen in the grain originates from N taken prior to enthesis and then remobilized later in the system. So we got to have that plant loaded up as best we can before we pollinate. And here's the rule of thumb. Because the flag leaf is so important in terms of the end filling, one of the measures of determining if you have enough nitrogen in your plants to produce good storage proteins is looking at the percentage of nitrogen in the flag leaf. If it's about 4.7 or greater, that should give you a nitrogen content of about protein content of about 13.5%. If you're really low in nitrogen in their flag, then you're probably looking at a reduction in your protein, and if there's a protein premium, you may want to do something about that. And you can do something about that about the time we're going to be talking about grain fill, right after pollination or slightly before pollination, depending on your time frame. And the last thing I would say is that the proteins that are built from those amino acids that are extracted from the entire plant are storage proteins. We know as gluten, which give you bread quality, and gluten is made up of two proteins, gliadin and glutenin, and those are the ones that cause people with allergies, the problems. But we can't sort of do much about that other than try and produce more something that isn't quite as intense as that in terms of the gluten content. Now, the storage all takes place in these amyloplasts. And these amyloplasts are cells that are specifically made, because they're called amyloplasts, for storage amylose and amylopectins. And that's what we got to build. Now, I found this, and it was very interesting because I've never seen it before, but everybody sort of talks about how plants can't mobilize copper and manganese and zinc and so on very effectively. Well, this study, which was sort of a synopsis of all the work that could be found, showing how much nutrient is remobilized from senescing leaves. So they took a look at how much was there and then how much was gone, and they concluded that that was remobilized. So wheat, we can pull almost all the nitrogen, potassium, sulfur, phosphorus, magnesium, calcium, which doesn't move very easily. Now you can see that manganese is not remobilized, and we even know that, and calcium generally is not very remobilizable, let's put it that way. But when we get into senescence, late in the cycle of the plant is basically cannibalizing everything, breaking down those cells in the vacuoles and moving it. So it looks as though the plant is going to be able to move everything. And if you can take a look at barley and canola, there are some blanks here because they don't have the data on that, but it was a really interesting find. And this is what feeds the world. Cereals feed the world, and maybe you can see why. Wheat, you move a lot of nutrients, and biofortification of wheat is one of the ways to provide the general population in underdeveloped and developing countries, because there are micronutrient deficiencies that are becoming global epidemics, for lack of a better term. Things like zinc, which are been mined out of the soil because we're not putting enough back on a global basis, is becoming a human health problem. And this one is an old slide, and it's useful because the plant, in fact, this is the seed. So the plant, in fact, redistributes the nutrients going into various compartments into the embryo. And you can see that it's got a lot of zinc, it's got a lot of manganese, a lot of boron, a lot of phosphorus. And then it blends in those minerals into the endosperm as well. And you can see there's a tremendous amount of sulfur in the endosperm. And sulfur is really key for bread making, because it's the actual sulfur and the covalent bonds in sulfur that, when it's being mixed, hook together and form the baking quality, and allows for the air spaces from the yeast to be confined in a specific density. So the plant is really amazing in that it puts the nutrients everywhere in the levels that it wants, and man has managed to capture that and use it in things like bread making. Now this is grain fill, our product produced by Active Agri-Sciences. Nitrogen, as you can see, potash, potassium in there, because potassium is key in terms of moving nutrients. Sulfur, we know that the N to S ratio is required in order to produce a good protein, because you know, two of the amino acids, cysteine and methionine, contain sulfur. So if you don't have a good balance of sulfur with nitrogen, you don't get a good supply of actually amino acids which build the protein. And iron is always involved with sulfur in terms of molecular bonding with sulfur molecules. The other thing that's in there are some other amino acids and vitamins, but a cytokinin, an artificial cytokinin called keratin, is also included in this in order to expand cells and slowly reduce the senescence period of plants. So a product like this should go on early in the morning or late in the afternoon. Once it gets too hot, it's not very good to put on foliar sprays. The potassium is essential here for remobilization of food reserves. Potassium is involved in moving things. It's not incorporated into molecules itself and proteins and molecules. It's used to pressurize the cell. It's moved to drive roots. It's moved to expand cells, and it extends the grain filling duration. And both nitrogen and potassium are used to build new cells, and we got to build those new cells, the amyloplasts, for example, that are useful. And we just mentioned nitrogen and sulfur in terms of formation of proteins. There are some amino acids in there. There are 12 amino acids that are in there to help with stress management. And specifically, proline can be formed and is also included in that in order to mitigate abiotic stress and deal with reactive oxygen species. And kind of lastly, we want to extend grain filling, and at least 30% of harvested nitrogen is linked to post-anthesis, flowering nitrogen supply, and that's from things like grain fill and from the reallocation or mobilization of nutrients. And that's about all I'm going to say about grain fill. You can go to the Taurus website and take a look at the yield results. We find, almost to my surprise, that initially we thought that we might get a protein bump, but in fact we're getting a tiny protein bump, but we're getting much bigger yield responses. And I think it's because we can, with that later application, we can delay that senescence, provide some nitrogen, supply some sulfur, but I think it's the delaying in senescence, maybe 3, 4, 5 days. That's maybe all it takes to really enhance the quality of that seed. So we're through with that. Now a few quick pictures. Just so you know, I always ask this question after the head emerges, what happens? And everybody says it pollinates. Not so. You can see here the sequence of events. The head emerges, and then this peduncle, this thing, the last stem, starts pushing the head up. And it pushes it up at about 1 inch to 1.5 inches a day, and the hotter it is, the faster it goes. And once it's reached out, I'll put this in here, and the seed head has a spikelet here, and usually there's 3 or 4 seeds there, and each one of these seeds is called a floret. Now you can see here, this is a primary seed head and this is a tiller. This was a dry year. The middle of the seed in the primary head produces 4. You can see 4 seeds, 3 seeds, 3 seeds, 3 seeds, 4 seeds, 4 seeds, 4 seeds, 4 seeds, 3 seeds, and 2s, and this is all you get out of the tiller. So, for folks that are growing reasonable crops, you want to try and keep your crop looking at one primary hit and one tiller to maximize your productivity. Uh, and by the time this plant or this spike gets through filling all of these seeds, it has really removed the potential of this tiller, because these seeds are going to be really, really big, and I'll show you some more seeds in a sec. And then it pollinates after we get extension. Now, each spikelet that's going to produce 4 seeds sits on this node. So the plant has to move nutrients into the node. So it starts pollination in the middle of the head and then moves in both directions. But that first primary spikelet gets access to the most nutrients. That's why you get the largest number of seeds right there. As you move to in both upwards and downwards on the seed, it's getting less and less nutrients, and that's why you're only getting 2 seeds, because that's all the plant can actually fill. So that's a tip for you guys that want to use your own seed for seeding until you want to change varieties. If you don't use a pedigree variety every year, just clean it really well and go after the big seeds, and I'll show you those. So here's pollination, and you can see right quickly within 3 or 4 days, and you can see the anthers are still stuck in here, the seed starts to grow. And this is just a bag of juice. And you can see here, as it grows, we start getting a little bit of formation of some structure. And here's a spikelet. So you can see, as I mentioned before, this seed and this seed will be virtually the same every time, those two. This one will be smaller, and this one will be the smallest, because you can see these two were pollinated off, maybe 1 day apart. And this one maybe 1 or 2, and this was later. So these two get the most nutrients. As it forms, we start seeing this happening. These water ripe become early milk. Then it starts forming structure. And then the structure, all those amyloplasts are now formed. Now the plant is filling them, and you can see it's just getting bigger and bigger by filling those amyloplasts, because about 75% of the seed is endosperm. So you got to build that big starch component, and the plant blends in the proteins into those starches. Now there's about 3 levels of starch: small, medium, and large, so that the plant makes it for germination of that seed. So when the seed is germinating, it then has access to sugars for a long period of time, along with the proteins, as the amylose and so on break down that material to feed that young seedling. And eventually you get down to fully ripe when it's done. And you get a little shrinkage here because it loses water in the ripening stage. And this is just, moving into the early milk and then later milk stage. And then we get the full seed starting to form. And then when you look inside the seed, you can see there was next to no structure there. So that's the time to get that nitrogen, get that sulfur, get that iron, get everything into the seed right after pollination, before it starts forming the structure. Because once it starts forming the structure and the endosperm is there, the plant is then focused on not growing more of them, it's focused on filling them. And at this point in time, you can see that the embryo has been formed and is still not mature. Here we go. You can see that the seed is fully filled with endosperm, and there's just a small amount of embryo there and seed coat, and the rest of it is all endosperm, which starts to harden. You get to the, I forget which this is, I think this is the hard dough stage, where you can still see your thumb mark in it. Once it's dried, the glooms and the other components, the pellia and lemma, start to loosen up, and it's ready for harvesting. And the plant does this all using hormones, and it's programmed into its life. Here you can see some pieballs, so there can be some issues with it. The seed is ready, the crop is done. It's filled. You've done your job, and now the seed is ready to go for next year and go to the elevator. Thanks a lot, and I hope this was useful to you. I haven't done this before. Good luck, and I hope you get a great harvest. Take care.